Brilliant. Hello, everybody. Welcome to our webinar on removing the bottleneck from high throughput imaging. This is a collaboration between Teledyne Photometrics, Dotphoton and the Wyss Center. On camera, you can see the three speakers for today. So I'm Dr. Matthew Koze-Dunn from Teledyne Photometrics, and I'll let my colleagues introduce themselves, and then we'll make a start.
Everybody. My name is Jocelyn. I joined from the Wyss Center. I work there part-time in the microscopy facility and part-time in R&D project.
And I'm Marianne. I come representing Dotphoton. I'm product manager there. And yeah, today I will show you our newest product. So very excited.
Brilliant. So this is a webinar of three parts. First, we'll hear from Jocelyn at the Wyss Center. Then we'll hear from me about photometrics, and then we'll finish off with Ariane and Dotphoton. So if you have any questions, please feel free to put them in the Q&A. And yeah, without further ado, let's get started.
Okay, I'm taking control of the screen. And look for,
You're a little quiet, Jocelyn.
Sorry?
You're a little quiet on my end. I don't know if that's the same for everyone.
Yeah, it's a bit the microphone.
Okay, but can you hear me or?
Now it's better. Now it's a bit better.
Okay.
You might have to scream.
All right, I will scream. I will speak a bit louder. Okay. Okay, so I didn't get control of the screen. And may I also add a laser pointer or something?
Okay, I've given you control, Jocelyn. No, okay, that doesn't seem to be working. So I can progress the slides for you if you like.
Okay, thanks. So yes, as I said, my name is Jocelyn, and I'm here to introduce a little bit the light sheet imaging facility within the Wyss Center. So we are a facility. We try to provide infrastructure to accelerate the discoveries in 3D biology. We are located at the Campus Biotech in Geneva, and we have access to different kind of microscopes, high-end microscopes such as confocals, slide scanners, digital pathology. And our specialty is light sheet fluorescence microscopy, where we provide not only imaging services, but also sample preparation as well, like tissue clearing, the processing. And we offer also training to our users, so they can actually operate themselves the microscopes. And also we provide them access to the image analysis tools that we have in-house as well. So if you can pass to the next slide. We have 4 different light sheet microscopes that 2 of them are custom made. They have arrived at different times of the facility. So as you can see, column and mesosfilm, those are custom made, and they were built in-house by Stéphane Pagès, our director. And the first one, so the facility now has almost 8 years in operation. And the first one that arrived, it was CON. It's called Clarity Optimized Light Sheet Microscope, which essentially was designed in Stanford in order to be able to image whole samples in 3D, but at high resolutions, with compatible with certain clearing protocols like Clarity. A few years after, the Mesosfilm initiative in Zurich developed this Mesosfilm microscope, which has a similar architecture as the CON, but offers larger samples and lower resolution, but higher fields of view, so you can image way faster than CON. Then 2 commercial options. The ClearScope came after. It has a different geometry. So it has this kind of oblique illumination, so you can image from the top, and you're not limited by the size of the sample. You can actually move the plate in X and Y, so you can image very large samples. And in our case, we were focusing on human tissue with this one. And the very last acquisition is the Smart Spin, which is actually it's a whole pipeline that batch processes samples super fast, so you can image and process tissue, many brains at a single time. So this is the highest throughput of our microscopes in the facility. So if you go to the next slide, just to give you a little bit of introduction of if you're not familiar with 3D biology and why it's important. Essentially, 3D biology aims at imaging tissue and the whole architecture of tissue, understand how the different entities are connected to each other, so cells and neurons, for example. Because if you think about it, living systems, it's just information flowing across these entities, these different pathways. And this is very well illustrated in this example of the mouse colon. The nervous system around the mouse colon is called, in the popular culture, the second brain. And you can see the complex network of neurons that go along the gut. And through these neurons, you will have electrical signals and chemical signals that will tell the gut when to contract, when to expand, when to relax, when to secrete hormones or absorb nutrients of the food that is passing through the gut. This was imaged with our light sheet microscopes, in the case Mesospin and Colm, and you can see next to it some images as well of this network that was also used to inspire a tool within in-house in the project of NeuroGI at the Wyss, which aims at developing tools to interrogate the gut-brain axis. So understanding how these networks are formed in 3D is very important to understand also the dysfunction and the diseases that emerge in this kind of tissue, in the gut brain axis in general. So if you go to the next slide, there you go. So how the 3D biology process is fascinating. It started with very ancient techniques of making tissue transparent, and these techniques keep evolving every year. There's new protocols to make the tissue transparent. So what these techniques do is basically, as you can see, the mouse brain over there, it has an opacity, a color, and all the structures like lipids that create the effects like scattering and absorption, then doesn't allow a sheet of light like the one on top. Laser here, but it's okay. So essentially, you remove all that. You don't have that attenuation by the tissue itself, and you can actually preserve all the structures of the tissue. So if you're interested to understand how neurons are connected in the brain, or you want to understand how vasculature, like how the blood flows in the brain, you can actually mark with fluorescent markers these specific structures. And then with a sheet of light, you pass through that, and then you can image only that without all the other structures. So it's very specific. Now, to have a really high resolution, but being able to image a whole sample of the entire brain, you need this kind of geometry of the light sheet where you have a decoupled illumination and detection. So the illumination is basically a laser beam that is scanned up and down to create a sheet of light. And then you collect orthogonally, you collect with a camera and detection objective. So there are many geometries now emerging. And with this, you might have small field of view to have very high resolution. So you need to reconstruct the image. You need to take many fields of views, pile them together, stitch them together to form the 3D reconstruction. And that is why you need specific computational tools to handle all this data, to reconstruct all these images. And then cameras play an essential role for having like a really sharpness and good contrast qualities on these kind of images. These new cameras, they can synchronize all the active pixels with the position of the laser in the sample. So like that, all the out of focus light is rejected, some sort of compatibility, and this creates really sharp images. So if we go to the next slide, this on the journey of Alice, the lightship journey of Alice started with mouse brains. Basically, these are really amazing systems in neuroscience to understand behavior, to understand many diseases, treatments, all that. And the very first lightship, as I mentioned before, was Qual, which enabled to image subcellular resolution. So as you can see in the video, this was taking a 4x magnification. But you can still see like all the neurons, how they are connected to each other, the axons. But if we want to image these at even higher resolution, so higher magnification, it was taking, you have a field of view of 1 millimeters. So it was almost 10 hours of acquisition. Because, I mean, field of view of 1 millimeter, and then you have a mouse brain that is almost 500 cubic millimeters. It's a lot of data to handle. So depending on the usage, you would generate 15 gigabytes to up to 1 terabyte of data. So you can imagine for the pre-processing, like stitching, reconstruction, it would take hours, and then the transferring of this kind of data to the users who are doing experiments was even longer. So Mesospin came after, and it increased a lot let's say you can go to the next slide. Mesospin increased the throughput because it managed to, by reducing the resolution and increasing the field of view, you could actually image a full mouse brain very fast, in 2 hours, and sometimes some regions in 25 minutes. So depending on what you do, you could combine, for example, Mesospin for like a whole overview. You want to see the forest, and when you want to see the trees, like go into the very specific part, you could go to Chrome and then image a specific region. This was being done, for example, in the lab of 15, which is in the campus biotech. They're the experts in spinal cord injury reparation. They have a lot of clinical impact, and they have to image a lot of mouse brains because they test their treatments. They want to understand if the spinal cord fibers will grow after treatment or not. So it's still a lot of data, and when we were transferring the data to them, we were always saturating the server. So yeah, this is a lot of samples generated and a lot of tools that need to be tested for transferring data. If we go to the next slide, the Mesospin allows us to, beyond the throughput, could also increase the data, the sample size. And this was the very first time in history that human eyes were clear. It was done in Institute La Vision in Paris, where they managed to clarify human eyes and label them with fluorescent markers, so specific colors of the eye. But you can imagine, like, the human eye is like almost twice the volume of a mouse brain. So you can imagine already in terms of data. The person who was doing this was coming to Paris with the samples and then going back to Paris with a bunch of hard disks with all the data to come up. So we go to the next slide. There we go. At the Wyss Center, we are very interested in management and advancing research in neurological diseases. And of course, the holy grail for that would be to image entire human brains. This is when the ClearScope arrived at the facility. The ClearScope removed the constraint of the sample size width. And so now you can actually put very large samples like the human tissue section of Alzheimer's patient that you can see on the right of the image. But imagine that if you want to image at very high resolution, so you can actually count the number of amyloid beta plaques, which are markers of disease in India. You want to count the neurons, you want to count the vasculature, you need quite high resolution. And for these large tissue sections, all this, it was already 2.5 terabytes of data. So only for a physician, and the team in-house, they developed a really nice tool to analyze this kind of data very fast. But still, if we were to image a full human brain, this system would be turning for months, and then the data analysis would be turning for months as well. So all that time, the users cannot access it. So we need to think creatively as the sample sizes increase. So we go to the next slide. So yeah, so far the Alice ecosystem in these 8 years of operation, well, they have seen almost 200 facility users, 90 projects or different institutions that are all over the world, in Australia, in the United States, all around Europe. So it's a lot of data that needs to be transferred. Only internally in projects like the one at the beginning of the GAT and many other internal projects that we have generated 500 terabytes of data. So we need tools to really handle that in terms of even analysis. So if we go to the next slide. So in 2021, we started a collaboration with Dotphoton because they came with this amazing new creative technique to actually compress images without affecting the pixel information. So I will explain that later. But we were kind of worried that any kind of compression would affect, the quantitative aspects, because in biology they need to do a lot of analysis. They need to do machine learning prediction and supervised learning to predict certain features. So it's very important that the number of cells, the total volume, the surface areas are all preserved. And with this kind of compression, it was, and that is why, and if we go to the next slide, they published this in collaboration between Wyss and Dotphoton. We go to that one. And then we implemented this technology for our phone, because back then it was the system that was generated the most amount of data. And it's been running. It helped us to really save a lot of storage in-house and for our users as well. So if we go to the next, now we are in the phase of we introduced actually the new product of Dotphoton, which is not only compression but also transfer of data. And our first case study was kind of urgency because we were testing a new control system home. And so we were a bit delayed, and we had to image a lot of mouse brains for mapping the brain secrets of social learning in 10% in each. So we have to, each mouse brain was 350 gigabytes of data. So we needed to transfer this quite fast. And with the Dotphoton, we could transfer the NAS with our internal servers, but also with the cloud, and that accelerated a lot. So we go to the next. So this is just to end. So I think it will keep growing in terms of new projects. There are always new creative samples, very, very solid samples, and every time with new techniques of clearing, we need always bigger sensors, bigger faster panel and faster transfer. So I will give the floor to Matt to explain a bit the technology that enables us this beautiful images, and then I am going to explain the transfer. Thank you very much, everybody, and looking forward to the questions.
Brilliant. Thank you very much, Jocelyn. And yeah, sorry for any audio issues you might be having, but Jocelyn is also available for any questions that you might have if you want to pop them in the Q&A. So now we're going to talk about Teledyne Photometrics. As mentioned, new technologies are needed to enable this sort of high throughput, very large samples, high speeds, and large volumes of data imaging. So I'm just going to introduce you to who we are at Teledyne Photometrics first, and then take you through some of our solutions. So Teledyne Photometrics, we design and manufacture these very high performance scientific cameras for demanding quantitative research. The company's been around for a very long time, over 40 years, and a part of Teledyne since 2019. We constantly move the technology forward. So we developed the first scientific grade EMCCDs and the first back illuminated CMOS cameras, and we continue to implement on that as time goes on. So the main kind of imaging solutions we're talking about here are these back illuminated CMOS cameras. And the one used often at the Wyss Center and in the case study is the Prime BSI Express. And we also offer the Kinetics and Kinetics 22. As you can see here, I've listed their maximum speeds at full frame and their number of megapixels. So you can see that the amount of data you're getting per camera is going up quite significantly. But rather than look at the products themselves, the most important thing is to see what specifications that you can measure from. The ones that we need to answer the scientific questions that people have in order to do this imaging are sensitivity, field of view, and speed. And we'll take you through why these might be useful for your kind of scientific imaging. So the first one is sensitivity. Essentially, you can't answer the questions you have if you don't collect enough data to show your image. So we need to be quantitative by making these cameras very sensitive. And you can show this with a sort of quantum efficiency curve or QE curve. Here we show percentage over wavelength. And this is essentially how many photons of fluorescence the camera is able to convert into electrons, which is going to make an image. And you want this to be very high. The Kinetics cameras and the BSI cameras, our Prime series, all hit at around 95% at the peak. This is around this nice visible light area here. But increasingly, cameras are pushing further out into the UV and the near infrared as well. We have new dyes. The infrared has new you can penetrate deeper into samples. There's less scattering and so on. So there's a big benefit from being a high peak and also a broad peak like this. Some examples. Sensitivity is very useful when you're looking at live cells. If you blast a live cell with too much light, you can end up having some issues of phototoxicity. What you want is a camera that can get really good images with a very soft, gentle illumination. Or if you're imaging at very high speed, you're not going to have time to do a lot of exposure. So sensitivity enables this kind of highly sensitive live cell imaging. And we've got different options here. This is a 3D imaging of a cell system with ESCAPE imaging system from TU Dresden. And this is imaging of organoids. These are more complicated groups of many cells that form this unique 3D structure in Erasmus in the Netherlands. Again, both of these benefit from highly sensitive cameras. The next thing is speed. So you want your camera to be able to image fast dynamic samples. Maybe you have something live that's moving around. Maybe you have a very large sample that you need to scan and tile across efficiently, especially if you're doing things like mesospin or working with large samples like the eyes that we saw. And you also want to be able to capture any sort of functional activity. If you're doing calcium or voltage imaging, these benefit from being able to image very fast. So you can see here in the graph, you have sort of typical CCD and EMCCD technologies. These cameras were used a lot in the past, but they're always quite limited in their speed. And then if we look at, back illuminated CMOS cameras, you can see the speed is going up exponentially as sort of the electronic engineering of these sensors improve. So the Kinetics is an incredibly fast camera, and then you can capture things that you just weren't able to see before. So these fast cameras also need to be sensitive because, again, to image at 1,000 frames a second, your exposure time can only be 1 millisecond. So you need to be able to collect all that data. Your camera can't be fast without also being sensitive. And these cameras will continue to get faster as we develop new readout methods and digital interfaces improve as well. So speed will also generate more data. Some great examples here. We like looking at voltage imaging and calcium imaging because these really benefit from speed. The one on the left is 2-color voltage imaging. This requires at least 1 to 2,000 frames a second or 1 to 2 kilohertz recording to capture the signals between cells directly. And here you can see it's done in 2 colors across these fast dynamic samples. Another way to do this is you can do functional imaging, but you can do it across an entire volume because the camera is so fast. You can image a slice of calcium activity and then image it all the way through what is, for example, here an entire zebrafish brain. So you can just scan all the way through and do functional light sheet imaging in 3D. So again, you can really design more complex experiments now that your cameras are getting more developed, essentially. And the last one is field of view, or FOV. This is the physical size of the sensor. Typically, we measure it in millimeters from corner to corner. So cameras have been getting bigger and bigger as time goes on. CMOS sensors are manufactured in all sorts of smartphones and things like this, so you can make much larger sensors. And the larger the sensor, the more data you capture. So if we look at something like an EMCCD, they're often quite limited in their field. We can look at the Prime BSI Express. This is 18 millimeters across, like a typical scientific camera field of view. You capture much more information. And then the Kinetics 22 is 22 millimeters, and the Kinetics is 29. So you can see these cameras get bigger and bigger and bigger. You capture more and more information. You don't need to take as many pictures to cover a large sample. And importantly, you can still do all of this at a subcellular resolution. So you can image very large fields of cells or tissues or organisms, but keep a very high resolution at the same time. But of course, as you increase the sensor size, you also increase the data. So for example, the Kinetics is a 10 megapixel sensor, and each image will be 20 megabytes, for example. And some examples here of field of view being a benefit. We have people imaging these very dense, thick brain tissue samples. This is the monkey brain imaged in Vanderbilt School of Medicine. You're doing calcium imaging with this very large, very thick sample. You need a camera that has a big field of view, but can also do all the things that you need to do functional imaging, which is to be fast and to be sensitive. And it also pairs up very well with things like spinning disk solutions here, so you can image these things very dynamically as well. However, we continually make cameras bigger and faster, but this leads into what we discussed earlier, which is the data issue. So these cameras are increasingly getting larger and faster. And if we, instead of looking at frames per second or megapixels, you look at the amount of data your camera can produce, you go from about 400 megabytes a second with the Prime BSI Express running at full whack versus almost 5 gigabytes a second on the Kinetics, if you were to use the full field of view and the full speed, for example. So you can capture a lot more with a very big, fast camera, but we need something to make up for the fact that your data is going up exponentially, especially if you're going to do something like a long acquisition and you don't want any dropped frames at all. So typically this is the role of something like your digital interface. USB is very popular, and USB is constantly coming out with new versions, and the data rate improves all the time, but it is being outpaced by scientific CMOS camera data rate. So we are all the way up to USB 3.2. USB 4 is available, but the cables are a little too short to use in sort of a lot of situations. So you can see here, you're being outpaced by something like the Kinetics family because these cameras are much bigger and faster. So you have exponentially more data. Imaging experiments are run for very long times. File sizes increase to gigabytes, terabytes, petabytes, and so on. So it can be unsustainable to run these very large cameras for a very long time and collect data and store the data without any bottlenecks in the pipeline. So we need further solutions in order for data to be able to catch up with all the advancements that have been made in scientific cameras. And that brings me neatly along to our last speaker, who's going to talk about how to remove this bottleneck from high throughput imaging on the data side. So over to you, Ariane.
Do I have control? Let's see. Did it work?
No, for some reason I can't give mouse control, so I'll have to advance the slides for you.
Do you think maybe because I have a few animations, maybe I have my screen ready to share. Maybe that would be easier.
Yeah, if you want to share it on your end, that's fine.
Yeah, I think it's gonna be easier. I'll
Cool.
just quickly.
We'll just switch over to Ariane.
Can you see it now?
Yeah, I see that.
Okay, smooth. Thank you. So today I'm here to show you our latest product. So I'm representing Dotphoton. Dotphoton is a Swiss startup. We're based in Zug, which is actually very close to Zurich, for those who don't know, here in Switzerland. After these great introductions by Teledyne and Wyss Center, and now it gives me, let's say, an easier way to present our product. So nowadays we're in very exciting times in research because, as we have seen, we can now acquire with very fast cameras at very good resolution, very high quality, and research also is evolving in such a way that scientists such as Jocelyn here in the picture can acquire longer movies. It's no longer time frames of hours, but now we're moving into days or weeks even, and that then of course results in big data sets that need to be analyzed, stored, transferred, and of course shared for the purpose of collaboration. So basically, as we saw before, let's say a typical now time-lapse size, it's in the order of 2 terabytes, which is already quite big, although as we saw before, it can get even larger. And to give you some ideas, to transfer a 2 terabyte time lapse in a 1 gigabit connection, which is quite common nowadays, you can even have faster, but 1 gigabit is, let's say, the typical. It will take around 4 hours. And then usually when you have this data, you end up transferring to different storage systems, as we will see later, maybe because you want to analyze it, because you want to have a backup, or also because you want to keep some intermediate place to save the data before you transfer it into, let's say, more storage that will last forever. But then this also means that your data is scattered in different places. So then that means that even though you have very good and amazing time-lapse movies, then what you have to do next can be sometimes a bottleneck. And today we're going to see how we can somehow fix it or improve it in some way. So if we look a bit more into detail on the workflows when you're acquiring, let's say, these large data sets, maybe it will resonate with some of you. So the first step is that you go to your microscope, you acquire your images, and then you save them in the microscope PC. And there you would save your large data sets, which are in the order of terabytes. But what means is that your SSDs, where you're typically saving your data, fill up pretty fast. Then after that, the next step most likely will be to transfer the data into some storage. It can be, for example, some intermediate storage that the facility provides so that you can save the data there and connect to some analysis workstations. You maybe also want to send a copy already to, let's say, an institute server or to your lab storage so that you can also save it there for later, for analysis or for raw storage. And then, of course, this transfer is quite slow because it's very large data sets. And then you have the issue that the more images that you acquire, then, of course, you consume your storage pretty quickly. Plus, if you indeed send your data to some analysis workstation, to some intermediate storage, maybe a hard disk, maybe some other storage in your lab, then you end up with multiple copies of the same file scattered around different storage systems, and it's not easy to keep track where they are. Finally, but not least, then of course you would like to share this data because you are in a project that you're working with people from, let's say, different countries. But this is not easy, and we've heard a lot about sending the data via post in a hard disk. And of course, this takes a lot of time and it's not very eco-friendly. So basically, here are some bottlenecks, but how do these translate to consequences in your imaging workflow? So these basically translate to delays in starting new acquisitions. So for example, if you're using the microscope and you're saving the data in an SSD and you are filling them up pretty quickly, then the next user has to wait for you to move the data out before they can start. Then if you acquire a lot of data, then you might need to buy some new storage, or you'll be forced to delete some data because you're running out. Then you also need to wait longer to start your analysis because you have to wait for the entire data sets to transfer for the workstation so you can begin analyzing them. You might have redundant copies in different storage locations, and it's hard to keep track of them and how to effectively delete them, for example. And then, of course, it's not easy to share the data, so this reduces the collaboration. So now that we see these consequences, here we go back now to our workflow. Before I explain to you a bit what is Jetraw and how does it would fit in your workflow, let's see how it will help improve these bottlenecks. So first of all, it will make your data sets smaller. So if we're talking of movies in the order of terabytes, they can go in the order of gigabytes. And this means your SSDs will be free for longer. Then transfer to your storage systems will be faster, and the same from the analysis workstations to your other storages. Then, of course, if the images are smaller, then that means that then you can increase your storage capacity, meaning that you will fill them more slowly. And finally, you will have a you can keep storing your data in different storage systems, but you will have one single access point to all the data. So this way, it will be easier to know where the data is stored, and you will only have to access it through one simple place. And then finally, data sharing also becomes easier and without the need of having to use hard disks in many cases. So what are the consequences then of using Jetraw? You will be able to acquire continuously without being blocked so easily by the SSDs getting filled in your microscope PC. The storage costs will be lower because it will take longer for you to fill them up, and hopefully you will not have to delete the data as one of the first options. You will have centralized access to your data, which also hopefully helps reduce the amount of duplicates of the data. Data sharing becomes easier, boosting collaboration, and the transfers are faster. So now that we saw this, you might already have an idea of what Jetraw is. So here is this is like a screenshot of our user interface, but we also have a command line interface and a Python SDK, but this one is more visual to show. So the way that Jetraw works, it's the middleman between your acquisition and your storage, and then from your storage to your analysis. And it's like a classical file system, so it should be easy to use. And the idea is that then you upload your data through Jetraw, so you drag and drop as you would send the data to any folder that you typically work with. But in this case, when you upload data through Jetraw, it will be compressed on the fly, so without the user needing to do anything else, so that the transfer happens with a compressed file and it will reach your storage compressed. Then you will be able to see the files in the file system. And the same way, when you want to then access this data, you will be able to download it. The data will be transferred in a compressed way, and then only when you are going to read it, let's say, it will get decompressed. The idea is that as a user, you will never see a compressed file, and this facilitates your workflow because you only need to work with the file in a decompressed way. Then you are able to connect to any storage system that you have. So we don't provide storage. So if you're working with a NAS, if you're working with any cloud provider, we can connect. And the idea is that you will see only one file system, but you can have the data physically stored in different places. But it will be easier because you can have full track because it's one single access point instead of having to connect to each individual, let's say, storage system and check the files. We also allow you to organize the data in what we call spaces. They can be arranged by labs, by projects, so that it's easier for you also to keep track of the different data sets that you have. And then we also have, let's say, a way for you to keep track of all of the transfers, how long they take, how many files are missing to transfer, so that you can keep an eye of the operations that you have going on. Also, as I was mentioning before, it also facilitates sharing. So you can actually manage the access for your spaces, and then you can share as easy as a link to your colleagues so that they can have access to Jetraw, and then they can download the files. Of course, if it's done via some cloud provider, it would be easier. Otherwise, if they have access to the storage, this can also be done via the link. So basically, if we go back to our researcher who is now acquiring very large data sets with very fast cameras, now they can be very happy because basically, if we go back to the 2 terabyte size movie, now we end up with 250 gigabytes, which is easier to transfer and quicker. So instead of 4 hours, now we take 30 minutes in the same connection. With the type of data that we see in life science, we achieve compression ratios in the order of 8 for this type of data, meaning that the storage and the transfers will be correlated to this compression ratio. And then finally, you can keep storing your data because we know it's not easy just to disappear all of your storage systems and use only one. You might have some legacy NAS or the intermediate storage systems from the facilities, and maybe now some new cloud storage, and you can connect to all of them, but this time through a single point so that it's easier to keep track of all of the data. So with this, I want to thank everyone who attended the webinar, and we're all happy to answer any questions that you may have. Otherwise, thank you very much. And yeah.
It looks like we do have a question in the chat from Bed Sharma. What are the compression and decompression speeds? I think that's one for you,
Yes.
Ariane.
Yes, so basically the compression and decompression happen almost at the same order, and it's 200 megabyte per second in one core, and typically, let's say nowadays even a very simple laptop has at least 4 cores. So we typically compress and decompress at the gigabyte per second, let's say, order of magnitude.
Brilliant. Does anyone else have any other questions? This meeting has been recorded if anyone wants to watch it on demand, I should mention. Or looks like we have a follow-up question. Do you need specific hardware to reach these speeds?
No, actually, it's run. We run it on CPU. We haven't had the need to run it on a GPU, mainly because it's quite fast, and typically the actually reading of the file takes longer rather than the compression or the decompression. And then the follow, is there any data lost during the compression? So basically the way that our compression works is that it only works for raw data. That's why the speeds for, let's say, transferring from the microscope are faster than from your workstation, because typically that data is already compressed, and therefore we cannot compress with the same compression ratio. So as I was mentioning, it's for raw data, meaning that the way it works is that we have partnerships with the camera manufacturers, and we obtain a report which is called the EMVA 1288, which ensures that the cameras, let's say, acquire within a specific range, so that all the cameras have a quality within their parameters. And this is the information that we use to do the compression. So it is not lossless in the sense that it's bit by bit accurate. So if you subtract the 2 images, it's not going to be black, but it's going to give you the same result as if you subtract 2 consecutive raw images. Let's say if you go to your microscope and you acquire within milliseconds the same sample, what you will get if you subtract them, it's basically the residue of the camera noise. And it's the same that would happen with Jetraw. So what we can say is that we ensure that it's a raw quality, but it's not the exact same identical bit by bit as the original. Can it be real time? Yes, actually, we have this in some systems. For example, the Viventis light sheet microscope already includes our compression, and while you are acquiring, you can compress with Jetraw on the fly, and it doesn't change your acquisition setup.
It looks like we have a follow-up about file types.
Yes, so we support. So yes, just for in case it is not clear. So Jetraw is it's a codec. It's not a file format. So we indeed can support any file type. It's just that it's a bit on demand. So we use we support the ND2, CZI, VSI, TIFF for now, and DNGs. But yeah, it can be possible to support more.
And is the induced noise statistically defined, Poisson Gaussian?
Yeah, so basically the idea is that if you indeed subtract Jetraw minus the raw and you want to analyze the noise, you will see that it's statistically the same as the one that you find in the camera, because we use basically the parameters from your specific camera to do the compression. So statistically, it's as if it's indistinguishable from another raw image. Yeah.
Perfect. And then we have one from the Q&A side. Do I need to install software on the server? Where does the compression happen? If I move the data out, is decompression done on the server?
Yes, so basically, wherever you install Jetraw, that's where the compression will happen. So for example, if I install it now here in my laptop, the compression will happen locally. And this is because the idea is that when you transfer files, the data should transfer already compressed, so that it's you can benefit from the speed. But let's say if you wanna, you can also do it. For example, what we see is that if you work, for example, with a cluster, an HPC, you can install the command line or the Python there, and then you can basically load the data as you would typically. But the data will be decompressed in the server if you have Jetraw installed there.
Oh, perfect. Thanks for all these questions, everybody. If anyone has any other questions, feel free to pop them in the chat because we probably have time for a few more. If not, then yeah, we'll be available on demand. And if you want to hear any more about this, then please feel free to reach out to us. We're always happy to take more questions. And yeah, thank you all very much for attending.
Thank you all.